Hi, my name is Sanchari Ghosh, and I am the content specialist at bioRxiv, Cold Spring Harbor Laboratory. And I'm honored by the opportunity to talk to Dr. Ulrich Schibler today, who is the professor emeritus from University of Geneva, where he dedicated his career to understanding the regulation of circadian rhythms. Thank you so much for joining us today, Dr. Schibler.
It's a pleasure. Just call me Uli.
Okay, all right, Uli. So your advent into the field of circadian rhythms was a little bit of a serendipity, right?
Exactly.
Would you tell us a little bit about that?
Yeah, with pleasure. So in the 80s, 90s, we were working on transcription, in particular tissue-specific transcription. And those times, there were only a few transcription factors known. I remember SP1 was the first one discovered by Bob Teachin in mammals.
Right.
So we were all searching for new transcription factors. And I had a postdoc in the laboratory from Canada called Chris Muller. And he used the phage expression library to discover, possibly discover transcription factors that bind to an element of the serum albumin promoter. Serum albumin is probably the second most boring genes. We worked on the most boring genes before, which was alpha amylase. But those days, everything was new, no matter what boring gene you were choosing for as a subject of study. So he found a protein that was binding to one of the elements called the D element. So we call it dbinding transcription factor, dbinding protein, DBP. And this went all well. We got a paper published in Cell, and we're all very happy. My grant was renewed. He got a job as an assistant professor at Queen's College in Canada. But after he left, there was a PhD student, Jerome Vieira, who was a son of a farmer. And the reason I say that will become evident in a second. So the student had to pursue this project, and with all of his efforts, he could never find a trace of this protein, nor of its messenger RNA. Although Chris had a plasmid, and I looked at the data, and I could imagine how this was possible. Because the student was very talented, one of the top students in biochemistry as an undergraduate. So after a few months, he came to me and said, this is all a phantom, it doesn't exist. And we were quite worried. So I went to the laboratory. Those were times where I still could use scientific equipment other than the telephone. And I went to the laboratory and prepared some rat extracts. And he did find it in my extracts. So it was a bit more complicated than just cheating. Or the other possibility was there were two cheaters in the lab, the postdoc and the professor. But fortunately, that turned out to be wrong. So we sat together, and Chris left about 40 extracts that he made when he was still in the lab, and Cheryl, the student, tested all of them, and this was completely erratic. Some of the extracts had a little, some had a lot, but none of them had exactly the same. And mine was somewhere in between, it was only the average of whatever he found by Western blot. So this was very strange, because if you take any other genes than known or any other protein, you find more or less constant expressions throughout the day. So we thought that perhaps one factor was time. Why did we think so? Because Chris, as many North Americans, came into the lab late, and then had a lunch, and then he started working and killed the rats in the afternoon, and then worked into the night. And that's why I said Charles was a farmer's son, because he had to milk the cows, to tend the cows early in the morning, and then he was in the lab at 7.
Right.
And by the time he prepared his extracts from the rat livers, this protein was just not there.
Right.
So then he did an experiment where he took samples, like he sacrificed 2 or 3 rats around the clock every 4 hours.
Mm-hmm.
We couldn't believe what we saw, because this protein in rats is oscillating more than 100-fold, so you just don't see it in the morning.
Wow.
Then, of course, we looked at the messenger RNA, same thing, the messenger RNA is maybe 2 hours advance or so, cycled also about more than 100-fold. And then we looked at nascent transcripts, and they also cycled. So this gene was transcribed in a highly circadian manner. That's how we started.
Wow. That's a great story. And since then, you have made some really seminal contributions to the field. If you had to pick 1 or 2 that are really close to your heart, or that you thought were the most surprising to you, which ones would that be?
Well, number 1 was the discovery of another postdoc, Aurelio Balsalover.
Mm-hmm.
And he worked on cancer before.
Mm-hmm.
So when he looked at the cells, he came to my lab and he looked at the cells. And this was late 90s, just after the first genes had been discovered. One of them was PER1, another one was PER2. And he looked at PER2, and it was expressed in these fibroblasts he was working on, rat fibroblasts. And then he looked at PER2, it was also expressed. Then we looked at all the ones that already had been discovered, and all of them were expressed. So then we looked at the literature, and we found that one of the immediate early genes, not a clock gene, but the immediate early gene that's induced in many situations, which is CFOS.
Right.
So he knew that CFOS you could induce very readily by adding serum to cells.
Right.
So we did that, and to our surprise, the curve went up, and then DBP went down. That wasn't the discovery of the cycle, because he only did an experiment maybe for 8 hours or so. But then he thought maybe it goes back up again, and PER back down again. So he did a 3-day experiment. It was just wonderful. These genes were cycling just as in the suprachiasmatic nucleus, because he managed to synchronize the cells by just adding serum.
Yeah. So these are cells that have not been in vivo for a long, long time, have been in culture for generations, and they still...
For 40 years at the time.
Wow.
Now it's 23 years more. No? But then when he discovered this rat fibroblasts, they were not in contact with the SCN for 40 years. So it was unlikely that they had a memory of 40 years.
Amazing. Amazing. Just tells us how inherent this whole mechanism is for a cell even.
It tells you another thing, though, which is much more general.
Mm-hmm.
And this was alluded to by Gary Rufko.
Mm-hmm.
I think biology is not hypothesis-driven. It is experiment-driven, discovery-driven. You could never... The systems are so complex. It's not like physics, where you can take a piece of paper and a pencil and write down the equations for quantum physics.
Absolutely.
You just have to do experiment. Just do a lot and see that you're not missing something.
Right. Yeah. Controls and keen observations. What did we do differently, right? Yeah, amazing. And then you spent a lot of time understanding the role of SCN in training the peripheral clocks that exist in the body. And so how you saw that field grow, right, over a period of years. So what is your main impression of that relationship between the SCN and the peripheral clocks?
Yes. So we believe, and I try to show that in my talk, that feeding cycles are the dominant timing cues or zeitgebers for peripheral tissues, for gene expression in peripheral tissues. And feeding fasting cycle, of course, are very general. We still don't know completely the metabolites and hormones involved. There was a paper by O'Neill who claimed that it may be insulin or insulin-related growth factor that play a role. But really, to be honest, we still don't understand everything here. But we know it's the most dominant site gainer, and we did this for several tissues, and we did it recently by in vivo recordings. So how are they regulated through the SCN? It's very indirect because the SCN drives rest activity cycles. And of course, the mice or rats don't eat when they're asleep.
Right.
So through the rest activity cycles, the SCN also drives feeding cycles, feeding fasting cycles. I say feeding fasting cycles because it's well possible that fasting is just as important in synchronizing these clock feeding. So this is very indirect. And then we looked at other signals. Maybe I should say that Aurelio, after he published his paper in Cell, looked at every signaling substance that we had on the shelf. And most of them synchronized it. So this was very surprising. You know, we had glucocorticoid activators of the cyclic AMP pathway. Whatever you tried, it worked. In one way, that was interesting. Another way, a little bit disappointing because you know, if it's everything that is doing it, what is doing it in vivo?
Right.
So we noticed that glucocorticoids are very potent synchronizers. And we also noticed that it is complicated because when you invert feeding cycles, for example, glucocorticoid, but not that much. And then later on, we also found through a screen for transcription factors that are immediate early transcription factors. And we found something else unexpected, which was a serum response factor, whose coactivators are MRTFs. So this was a very talented grad student called Alan Gerber, and he found that every day the actin skeleton oscillations changes. So you get a lot of these fibers, you know, during the night, and nobody has ever seen that because nobody looked at it. So it's actually there's a protein. We don't know what that protein is, but we know it's a protein in blood. It's the actin skeleton. The actin skeleton regulates SRF, and SRF is probably the strongest inducer of PER2 and synchronized PER2 of the serum factors that we and others analyzed.
Right.
So both glucocorticoid and this protein that is apparently cyclically regulated by the SCN are not following feeding rhythms. So you have pathways that are more directly connected to the SCN.
Right.
And you can test that now with our recording device, because you can record rhythms. That's just a normal intact mouse that expresses a reporter gene either in all cells or just in the liver.
Yeah.
And now you can, after a few days, you can switch to an inverted feeding fasting cycle.
Right.
If you look at the normal animal, at an intact animal, it takes 8 to 10 days, or perhaps even a bit more, to completely reach a new phase.
Right.
If you take a mouse that has been lesioned for the SCN, no SCN, then it takes 1 day. So it seems that the SCN is slowing down. It sends out signals to slow down. It wants to, you know, it wants to be the master, so it wants to keep everything under control. And if you remove it, then the feeding cycles change it immediately.
Right.
So I always call, and maybe I don't know whether I should say this is politically correct. I always call the SCN, it's like a Catholic priest. Do what I say and not what I do. Because it turns out that the SCN actually lacks the receiving receptors or whatever for everything it controls. It's the only tissue I know that doesn't have glucocorticoid receptor.
Wow.
It is not responsive, as Joe Takahashi shows, to HSF1, to the temperature cycles that also synchronize, perhaps unexpectedly, circadian rhythms. So it sends out signals, but doesn't listen to them itself.
Yeah, so it doesn't take feedbacks, it just gives out the orders.
It doesn't take feedback, at least not to its own signals, yes.
Right. And it was surprising to see the phase coherence in the independent hepatocytes in the SCN lesion model, right? Like from the data from your talk. And I was just wondering, given the emergence of how important the understanding that circadian rhythms are so important in regulating metabolism. We've had several talks this past week on that topic. What do you think is the impact of this finding from your data on understanding metabolic diseases today?
I have to be very honest. I mean, the question is, do you synchronize an organ on the organ level because it has crosstalk between its own cells, or do you synchronize every cell in the organ? Well, our data and those of Joe Takahashi published earlier, based on in tissue explants, that he analyzed in vitro, indicate that there is some phase coherence in many organs. And you can ask the question, does this have an advantage? Because the mouse does not foresee that you will SCN lesion it, no? It is not selected for. So, of course, you can always come up with hypotheses, and for a big organ, it may be easier if you have both signals from the SCN and signals from within the organ to reach good synchrony. But I don't think it's the only way you could do that.
I see.
You have to understand evolution. If something is there, it either is useful or it doesn't hurt.
Right.
And a lot of these things may be based actually on design principles rather than on importance. I really don't know what the phase coherence within the liver, what that contributes to driving cyclic metabolism.
Right. Okay. And I have one more question for you. So if given the current state of the field and how much it has evolved since the time you started the lab, if you were to start your lab today, what would be the one burning question you would pursue to understanding circadian rhythms? Well,
I frequently thought about this. I mean, we didn't start the lab, of course, on circadian rhythms. The lab on the tissue-specific gene expression, that was in 78, a very long time ago.
Right.
And we stumbled into this by complete serendipity, as you said, and I told you the story. So, what would I do now? There are two things I probably would start. One important question, really basic question, that we don't have complete or even incomplete answers is, why is germline DNA immortal? So in other words, we say that, there are aging theories that say that DNA gets bad with age, gets mutated and so on, but apparently that's not true for the germline DNA. And the reason, one of the reasons why I'm interested in this is because that's an advantage of being old. Young people don't remember these experiments anymore. But one of my scientific heroes is Joe Gall. And he studied lump brush chromosomes. So you find that, for example, in the frog or normally in amphibia oocyte development, you find that all DNA is transcribed, not in units of genes, but in units of loops hanging out from the central filament of the chromosome. These, since they look like a lump brush, they're called lump brush chromosomes. So these transcripts are useless because they're not, they read through genes in sometimes in opposite directions. Right. And the question is, why? Right. So So my hypothesis is that it's actually not the transcripts are important, but transcription coupled repair. Because transcription coupled repair must be very efficient because it repairs genes, though.
Right.
So I believe now it says this, but how about mammals? Do they also have one brush chromosomes? Perhaps not, but they also have like a, you know, 100-fold higher transcriptions in these oocytes. So maybe the organization is a little bit different. Now we have probably had a postdoc, Ed Schmid, who is now professor in Montana, in the United States, and he didn't want to work on circadian rhythms, so he worked on testis transcription. He found that the general transcription factors and polymerase II are 100 to 1,000 times more abundant in sperm during sperm cell development than in other tissues. I think it's the same. I think it's a proofreading mechanism for germline DNA. So the hypothesis is that you have proofreading, very efficient repair before you pass on the DNA to the next generation.
Right.
Perhaps also in addition, there are quality proving mechanisms. For example, why do females only make one egg out of 4 cells? Whether you make division, you have these 3 posters, only one, and maybe if one looks at the DNA, there may even be papers on that. I didn't check the literature. But the DNA, it selects the best of the 4 cells.
Right.
And you could imagine that the ones with good DNA have stronger mitochondria, so they move a little faster, you know. It's all hypothesis, but I think this is a very general, very important question that has not been solved to my satisfaction.
Okay.
So that's one thing. And the other thing I would perhaps do is to just take genes that have no resemblance to any other genes. So you look at the gene banks, and you take like 20 genes that have not even resemblance to other proteins. And then study, you know, try to study what they're doing, because there you have new land and you're bound to make an important discovery if you do move them. So these are the 2 things that will probably start.
Great. Well, thank you for joining us today again, Dr. Schibler. It was wonderful talking to you. Thanks a lot.
My pleasure. Thank you very much.
Thank you very much.